Today, you are going to hear about the extraordinary collaboration between families, clinicians, and scientists around the world that led to the identification of a new rare genetic disorder, the first ever linked to a long non-coding RNA gene. The discovery from Northwestern Medicine, the Broad Institute of MIT, and Harvard University scientists was recently detailed in the New England Journal of Medicine and highlights the need to examine non-coding parts of the genome when diagnosing rare diseases. Northwestern Medicine investigator Dr. Gemma Carvill is my guest. She is the corresponding author of the study and an assistant professor in the Ken and Ruth Davey Department of Neurology and of Pharmacology and of Pediatrics at Northwestern University Feinberg School of Medicine. Her lab's goal is to determine the genetic causes of epilepsy and how pathogenic variants in these genes affect how the brain develops and functions. She joins me today to talk about this important new discovery and how it came to be. Welcome to the show, Gemma.
Thanks so much, Erin. I'm excited to be here.
Well, we are very lucky to have you. And today we are talking about your involvement in this New England Journal of Medicine study that began after the family of a little girl named Emma Broadbent reached out to you after struggling to get an accurate genetic diagnosis for Emma. And Emma lives with severe delays in brain development and a host of other health issues. So tell me a little bit about Emma, her condition, and her family's outreach to you.
Yeah, Emma is a fantastic little girl, a spirited little individual for sure. I first met Brian, her dad, in person actually only very recently, but on the interwebs a couple of years ago now. It all started back in 2013. So we first identified that pathogenic variants in a gene called CHD2 could cause a neurodevelopmental disorder. But kids who had pathogenic variants in the CHD2 gene generally presented with very bad seizures, and some of them can have neurodevelopmental delays as well. But what Brian had found through collaboration with the UDN, which is the Undiagnosed Diseases Network, he had been working with them for many years to try and find whether there was a genetic variant that caused Emma's condition. And so what they found was a pathogenic variant that was a little bit different to the other kids with CHD2 variants, in that this pathogenic variant wasn't actually in the CHD2 gene, it was just upstream. And so he immediately appreciated two things, which is incredibly impressive for an individual who hasn't been classically trained in genetics. He immediately noticed, one, that the symptoms that his daughter had were very different to the symptoms of the kids who have pathogenic variants in the CHD2 gene. So he immediately noticed the difference in kind of their clinical phenotype. The second thing he noticed was that those genetic variants weren't actually in the CHD2 gene. And so he was somewhat suspicious that this might be the cause, but really essentially sent me a cold email out of nowhere to say, my daughter has this genetic variant. She looks a little bit different to the other kids. What are your thoughts in terms of this being the genetic cause for my daughter? And so that was really where this all started, was that cold email many years ago. But Brian was then really instrumental in bringing everybody in the team together. And he was really the one that drove everybody, got everybody in the same room to try and answer this question of, could this genetic variant be what is causing Emma's condition?
Because CHD2, this is one of your genes. This is something that you study in your work to establish the genetic cause of epilepsy. Tell me about some of your previous work with the CHD2 gene and why it's known as this Goldilocks gene.
Mm-hmm. Yeah. So, back in 2013, we first identified these genetic variants in CHD2 and then linked them to this epilepsy phenotype. And since then, for the last, it's more than 10 years now, have been studying this gene and trying to figure out, when you have a pathogenic variant in this gene, how does it end up causing those seizures? And so as a result, we've also worked very closely with the Family Foundation. And so what tends to happen in these rare neurodevelopmental disorders is that when a family receives a diagnosis, so in other words, a genetic diagnosis that causes their disorder, families tend to coalesce around that single gene and put family foundations together and essentially do a lot of work finding other families who have mutations or pathogenic variants in that gene, setting up NGOs, connecting with researchers, doing fundraising for research. And so probably about 5 years ago now, we connected with 2 moms whose daughters have a CHD2 variant and established what we call the Coalition to Cure CHD2. And so this is the family foundation who organizes a lot of the outreach for families with CHD2 variants. So Brian connected with the family foundation through Emma's diagnosis and now helps guide kind of some research questions for both my lab, but other labs as well. But to answer your second part of the question is, why is it this Goldilocks gene? So patients who have pathogenic variants in CHD2, what ends up happening is those variants essentially destroy the function of that protein. And so we call that loss of function. And so in individuals with this condition, they have about 50% of the levels of CHD2 compared to the general population. And so they have too little CHD2, and this is what causes the seizure disorders. In Emma's case, she doesn't have a pathogenic variant in CHD2, but we found that she has a deletion just upstream of CHD2. And her deletion encompasses the long non-coding RNA called chaser. And the way in which we now think chaser works is this long non-coding RNA controls the amount of CHD2 that is made in the cell. And you can think of it almost as a brake. So the long non-coding RNA acts as a brake, and you kind of take your foot off the brake or put your foot down to control the amount of CHD2 in the cell. In the case of Emma, with the deletion of that non-coding RNA, what ends up happening is there's no longer that brake. And so now what happens in Emma's cells is that she has too much CHD2. And that's likely what underpins many of her challenges in terms of neurodevelopment, but also neurological function. And so it's a Goldilocks gene because both too little and too much is detrimental to brain development and function.
Let's talk a little bit about how you were able to determine this. You were able to bring Emma's cells here to your lab at Northwestern and grow them and study them. What were you looking for? And then again, what did you find?
So I spoke a lot about how both too little and too much of CHD2 is bad. Yeah. And so we knew from some studies in mice that if you deleted chaser, the long non-coding RNA, in mice, what happened was there was an increase in the production of CHD2. So what we thought was maybe this is what is happening in Emma's cells as well. So our original hypothesis or idea was to test if in Emma's cells she had more CHD2 than the general population. And so the way in which we did this was Emma had a tiny little skin punch biopsy. It's essentially just a tiny little 1 centimeter skin punch biopsy. And so she underwent that procedure at Baylor. You can then take that teeny tiny little skin punch and you can put it essentially in a Petri dish. And what ends up happening is that these tiny skin cells essentially grow out of that tiny little piece of tissue. And you can establish what's called a fibroblast cell line. And so that cell line you can grow many different generations, and you can keep those cells alive. And they're incredibly useful for answering some certain types of questions. But what we really wanted to do was take Emma's fibroblast cells and turn them into induced pluripotent stem cells. And so this is essentially taking its cell type and basically pushing it backwards into a type of cell that has the potential to be able to be differentiated into multiple cell types in the body. And so in other words, you can take that stem cell and you could turn it into cells that look like cells in the brain or cells that look like cells in the heart. And so it's a very nice model system where we can try and recapitulate some of the neurological features that we see in these kids using this iPSC stem cell model. So we did that with Emma's cells, turned them back into stem cells. Yeah. And then we addressed our original question is, what are the levels of CHD2 in Emma's cells? And what we found was that indeed, as with mice, in humans who have a chaser deletion, there's an increase in the amount of CHD2 protein relative to the general population. So we did that both with Emma's cells, but then also later There's another patient from France, and we did the same thing and showed there's basically a 1.8-fold increase in the amount of CHD2 in these individuals who have chaser deletions.
Well, and again, this discovery was made by looking into the non-coding regions of the genome, which has been understudied and mostly unknown to scientists. Why have these regions been so understudied, and what challenges do they present for geneticists, people like you?
Yeah. So we like to joke that the non-coding regions of the genome is basically job security because we basically have no idea what most of the genome does. And so I'll probably be very, very old and we still won't know the answer to that question, or the answers to that question, rather. To take a step back, what are non-coding regions? So if we look at our genome, about 1% of the DNA in our genome, it makes proteins. And so what happens is there is a gene that codes for an RNA, and then RNA makes protein. And so amazingly, everything that makes us human, or every single component of our cells, is essentially made from proteins. But it accounts for only 1% of the entire genome. The other 99% of our genome, we really don't know what it does. And so we have, in some regions, we have a good understanding of what the purpose of that DNA is, but by and large, we don't. And so what chaser is a long, non-coding RNA. And so chaser makes an RNA molecule, but then it doesn't make a protein molecule. And so rather, what that RNA molecule does is it's the job of the RNA molecule to control the amount of CHD2 protein that is made. And so we think that's really its primary function in the cell, is that you make this RNA and its probably one job is to control how much of one protein is made. And so there are tens of thousands of long non-coding RNAs in the genome. And I would say we know only a tiny percentage of the function of those long non-coding RNAs. Some of it, in the past, we haven't found cases like this because we haven't been looking. And so traditionally, if an individual has a genetic test for breast cancer, for instance, what the genetic testing company will do is look at all of the coding regions, so the parts of the genome that make proteins, look at the coding regions for genes that are known to cause breast cancer. But that's it. And so that is the sequencing that will be done. And some of that in the past has been a technological limitation in that we could only sequence those pieces. But now we have the technologies that are pretty cost effective to be able to sequence the vast majority of the genome. So we can look at the 1% and we can look at the rest of the genome as well. And so now we were able very easily to sequence the genomes of individuals. But the real bottleneck and challenge now is that interpretation, is if we find a genetic variant, or in this case, a large deletion of a chunk of DNA, we have no idea what the downstream effect, if any, that may cause. And so this is an example of how you can have a deletion in a long non-coding region that can have very profound impacts in terms of development and neurological function for these patients. It's by no means an isolated incident. And so we think that in the future, as more folks do genetic genome sequencing, we'll start to uncover more and more examples of this where non-coding regions of the genome can be implicated in health and disease.
I think there's about 30 million people with rare diseases in the US. About half of those are children, and they're thought to be of genetic origins. So you do think that these long coding RNAs and non-coding regions could play a role in these other disorders?
Almost certainly. Yeah. Not all of them, but I think a very large percentage of the we call them unsolved rare disease cases could almost certainly be due to non-coding genetic variants as well.
What kind of message does this send, this discovery, to your colleagues and other folks who study these types of diseases and are looking for answers for families?
I think one of the main takeaways from this is that we shouldn't ignore the non-coding regions of the genome, and that we really need concerted efforts to come up with a roadmap of what does the rest of the genome do. And this is not something that we've completely ignored as scientists. Scientists have been working for many years and trying to figure out what the non-coding regions do. But I think we need even more research is essentially the answer to try and figure out what these non-coding regions do, which ones are important in health and disease. and just in general development as well.
Now, you mentioned this story didn't end with identifying the genetic cause of Emma's disorder. There were two other children. You mentioned one in France with this exact condition. So tell me how this came to be that you were able to connect and basically diagnose these other children.
Mm-hmm. So about probably 2 or 3 years ago, we had done the confirmation experiments that showed that Emma had increased CHD2 levels relative to the general population. And so we had Emma's answer. We were pretty confident we had Emma's answer probably about 3 years ago. The big sticking point was if you have one patient, it's very hard to say conclusively that this is the cause of their disease. And so really we spent the last 2 or 3 years reaching out to collaborators across the globe, giving presentations in multiple different forums, so in human genetics conferences, at rare disease conferences, and essentially asking the world collaborative of rare neurological conditions to go and look at their data and see if they could find this deletion. And that was really how this discovery came about at the end of the day. It was via a conference where one of our colleagues was giving a poster and they happened to see it and go and look in their data and then found the second and then third individual who has a chaser deletion. Interesting, both of the other two patients are both from France. And so again, this just speaks to that we don't think that these are the only 3 individuals in the whole world who have chaser deletions. It more speaks to nobody's looking in the right place. But as soon as people start to look, that we find these deletions. And I think that's true for chaser, but I think it's true for many other conditions as well, is that we really just need to start looking in a concerted way.
I mean, you just mentioned there's so many different people around the world with different areas of expertise who contributed to this discovery. How important is this type of collaborative team science to study such complex genetic problems?
Yeah. So it's one of my favorite parts of my job and one of the most underrated and sometimes difficult parts of my job is it can sometimes be very difficult to get researchers in the same room playing on the same team. And I will say that Brian was really our cheerleader who put us all together and got us all in the same room across often 2 or 3 different continents on different time zones, kind of worked with everybody to bring everybody in the same room. But I think it really is a nice testament to how team science done right can really advance our knowledge of the genome and the types of genetic variants that can cause rare disease. And so we've been working on this, like I said, for 3-5 years to put this story together. And everybody kind of had their own part to play. Kind of the Broad and Baylor were very instrumental in the beginning in finding the chaser deletion. And then kind of my lab came in later to look at what happens when we delete chaser to the levels of CHD2 and kind of looking more in Emma's cells. And now moving forward, we are still collaborating. And so we're moving forward with trying to think of ways in which we can use chaser to control the amount of CHD2 as a potential therapeutic, as well as looking in Emma's cells and the other French patient cells, can we reduce the amount of CHD2 as well? So we call these gene targeting or precision therapies, where we're now trying to develop therapies for this very, very rare condition. And so we're doing that really in collaboration with Brian and his family and the other families, but also the Broad as well. And so it's a kind of an ongoing and I think a nice case study of how these team science approaches can really work.
You mentioned looking into these possible therapies. How could such treatments hypothetically work?
Yeah, so we know that chaser controls the amount of CHD2. And we know that in individuals with that seizure disorder, they have too little CHD2, right? And so in theory, if you could target chaser, reducing the amount of chaser, that might be a way to boost the amount of production of CHD2 in those individuals that have too little. And so the way in which we're doing that is we manipulate the amount of RNA, of the chaser long non-coding RNA, we manipulate how much RNA is made from that transcript. And so it's taking advantage of a natural process to kind of tinker with protein levels.
I mean, and it's unknown if a hypothetical treatment like this would be able to help Emma in her lifetime, yet her father still plays such a
Hmm.
critical role in moving this along. He's actually an author on this
Yep.
New England Journal of Medicine article.
Yep.
How essential are the Bryans of the world in pushing science like this forward?
Yep. I would say it's been remarkable how this has changed over my career. And my career is not that long, and that I kind of started in this field in 12, 13 years ago. And we didn't even know what the genetic causes were for these conditions. And now, one, we know that the majority of rare disease is caused by genetic variants, but also it's no longer just a scientist sitting in isolation studying a gene for 20, 30, 40, 50 years. Now it's very much a collaborative effort with people like Brian, with foundations like CCC. I would say that all of us that work in rare genetic disease, I would say that we spend a significant proportion of our time working with family foundations and people like Brian. And I think that that's a great lesson for young trainees as well, to understand that their science matters. It matters for the people behind the condition as well. And I also think it informs our science. We know what is important to families. We know what their struggles are. And we can tailor our science so that it best fits addressing their needs. And so I think that has really changed a lot over the last couple of years. And I think it's good for science. It's good for moving towards gene targeting therapies and getting answers for patients.
Again, your lab's focus is to determine the genetic causes of epilepsy. This could lead to gene targeting therapies to correct the root cause of this disorder. How does this new knowledge in Emma's case get the field closer to designing such therapies?
So epilepsy is the third most common neurological disorder. And in addition, 30% of individuals who have epilepsy are resistant to current medications. And so as a result, they have ongoing seizures that we do not have the tools to treat and to rectify. One of the ways that we're trying to rethink how to treat epilepsy is, instead of treating seizures, which is really just the manifestation of this condition, is instead to treat the root cause. And so in so doing, our goal is to, one, identify what is the genetic variant or sometimes variants that cause an individual's epilepsy, and treating that root cause as opposed to treating the symptoms of epilepsy, which is the seizures. And so Emma is an excellent example of this, in that if we can manipulate the levels of CHD2, we're not really treating the symptoms of the condition, but we're treating the root cause. And so by correcting this, the goal is to eventually have very precise therapies to treat rare genetic conditions. And so it applies to Emma, but it's really the goal broadly of the field of rare neurogenic conditions.
Tell me about the environment at Northwestern that really allowed you to pursue this. This was kind of a one-off case in a lot of ways. Tell me about the support that you had in order to make this science happen.
So while this is a very rare cause of a neurological condition, I think it speaks to a broader effort across neurology and across Northwestern to do more precision medicine type approaches in treatment of health and disease. And so traditionally, like in epilepsy, we treat the seizures, so the symptoms of the condition, rather than the root cause of the condition. I think we in my lab are starting to think about precision therapies and gene targeting therapies, but we are not operating within a vacuum. And so one of the really encouraging things and exciting things about being at Northwestern is that there is a very big movement towards this line of thinking, where we're committed to identifying the cause of an individual's neurological or other condition and treating patients based on their genetic makeup, as opposed to treating the symptoms of a variety of different conditions across the university system as well.
Is there anything else that you would like folks to know who are hearing this story for the first time and they're excited about this new approach, that there could possibly be precision medicine for people with diseases that this has never been an opportunity for them before? Any parting words as we leave today?
I think it's a nice example of the power of genetics and how having a genetic diagnosis can move you towards precision therapy. And so I think this is one of the most encouraging things is that genetic testing is becoming more and more available across the US and worldwide. And I think I would love to encourage people to think a little bit more about genetics in terms of your health and disease and the potential options that are out there in terms of a genetic diagnosis.
Gemma Carvill, thank you so much for sharing this really extraordinary story and the work going on in your lab. And we will continue following along as more discoveries are made.
And thanks so much for your time and getting the story out there. It's a very exciting collaborative story, and I'm really glad that the world is getting to hear about it.
Thanks for listening. Please click the bell to receive notifications about our latest episodes and follow us on social media at NU Feinberg Med to stay up to date with our latest research findings.